Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Plant Biology and Biotechnology conducted in the year 2020. So coming to the first question. One of the following is an instrument used to measure water potential of leaf tissues. So it is psychrometer that is used to measure water potential of leaf tissues. Answer is option A. The next one. The organism that is suitable for mapping the gene with respect to the centromere is. So it is Neurospora crassa that is suitable for mapping gene with respect to centromere. Next one. The stain used to test seed viability based on the pink color developed by the living seed tissue contains one of the following salt. So it contains tetrazolium salt. Answer is option B. Then next one. A cross was made in Snapdragon plant between capital A, capital B, small b into small a, small b, small b. What proportion of the progeny will be small b if the two genes A and B are 24 map units apart? So these are the parentals, that is capital A, capital B, small b, and capital A, small b, small a, small b. Now these will produce the gametes AB, A small b, A capital B, A small b, and this will produce the gametes A, B. Upon crossing you will get upon crossing you will get these progeny. So recombination frequency is 24%. So each recombinant will be, or each recombinant progeny, that is this ones, the center ones. These are the parental phenotypes AB, AB and small a, small b. They are the parental genotypes. The recombinant progeny will be 24%. So each recombinant progeny will be 12%. The parental will be 38% each. So what proportion of the progeny will be small a, small b, that is the parental progeny they have asked. So here it will be 38%. So answer is option C, 38%. Then the key switches in signal transduction circuits regulating gene expression are. They are transcription factors. Answer is option B. Removal of male organ from a hermaphrodite flower to prevent selfing is known as. It is called as emasculation. Answer is option D. The mating of plants in all possible combination is known as. It is called as diallele mating. Answer is option B. Then arrangement of petals and sepals in a flower bud before it opens is known as estivation. Answer is option B. When a cell is fully turgid, which of the following will be 0? So suction pressure will be 0. Answer is option C. Then next one. Hydatodes are water secreting glands. Answer is option C. The deamination of 5-methyl cytosine produce thymine. Answer is option B. 1 centimorgan is defined as the genetic distance between two loci with a statistically corrected recombination frequency of 1%. Answer is option C. All these are direct questions. Okay. Then, which of the following amino acid in proteins cannot be post-translationally modified? So it is alanine. The amino acid alanine and glycine are not involved in post-translational modification because they are not having functional groups in their side chains. So alanine and glycine, they cannot be post-translationally modified. Then reverse transcriptases. It is RNA dependent DNA polymerase. This is a usual question for many entrance examinations. RNA dependent DNA polymerase. Answer is option C. The next one. A plant species nearing its extinction due to virus infection has been given for tissue culture and micropropagation. Scientists below choose the 4 explants for culturing. So, which explants would have produced virus-free plants? So, 1 and 2, that is meristem and shoot tip, they would have produced virus-free plants. So, answer is option A, that is 1 and 2. Then consider the following statements on the common feature between phase contrast and dark field microscope. Both increase contrast between specimen and background without staining. That is correct. Which of the following is true? So 1 is correct. Both make specimen fluoresce on a dark background. No. Both make specimen appear dark on a bright background without staining. No. Both make specimen visible that refract light away from the objective. No. So the answer is A only, that is 1. 1 only. So answer is A is correct. That is both increase contrast between specimen and background without staining. Then next one. How would you measure the transcript abundance of a particular gene? So transcript abundance is mRNA abundance. So mRNA can be analyzed by performing northern blot technique. So answer is option B. Though DNA and RNA are nucleic acids, isolation of RNA in the laboratory requires extreme precautions and pre-preparations than isolating DNA. This could be because DNA content of a cell is more, but RNA will be present in lesser concentration and therefore care should be taken to avoid loss. That's not correct. Then RNA is smaller in size than DNA, so it requires stringent procedures to capture those smaller molecules. That is also not correct. There is nothing like that. Then RNA is unstable and prone to degradation. Yes, that is correct. But DNA is relatively stable, that eases DNA isolation. That is correct. So RNA is very much unstable when compared to DNA. So we have to be careful while preparing RNA for isolation of RNA. Then DNA is bound to proteins that safeguard the molecule, but RNA is naked, which makes the separation difficult. That is also a wrong conception. So the answer is option C, that is 3 only. Then identify the mismatch. Zooxanthellae, Pyrophyta, Cephalurus, parasitic, Eulothrix, branched filaments. They are not branched, they are unbranched filaments in Eulothrix. So answer is option C. Then, to survive harsh environments, plants have acquired several adaptations with specialized functions. Below is the list of traits A and their potential role, that is B. So waxy cuticle restrict water loss. So P3. So A and D can be not taken into consideration. We will see for C and B. Thick or lignified cell wall, that is for mechanical support, Q1. So option B will be correct. Anyway, we will see the other options also. Homeohydry, so that is vascular system, and pigmentation, protection against excess light. So option B is correct. P3, Q1, R4, S2. Then plankton will be absent in. So plankton will be absent in case of lentic biome, lotic biome, marine biome and lakes. Plankton will be absent in lotic biome. Then next one. The most commonly used stain for producing bands on chromosome for karyotyping is. So it is usually the Giemsa stain that is more commonly used for banding studies in chromosome. Then next one. Cistron is the genetic unit of biochemical function. Answer is option B. Then winged bean is Phosphocarpus tetragonolobus. So winged bean is option D. Then the feature unique to monocotyledonous root is, it is polyarch xylem. So exarch is the arrangement in which the protoxylem lies outside and metaxylem lies inside. So that is called exarch condition. So the feature in monocotyledon's root is it is polyarc xylem. The next one. Which of the following statements about keratin protein of hair, wool and nail is false? Exhibits alpha helical structure. That is correct. Is a fibrous structural protein. That is also correct. Exhibits stability due to hydrogen bond and disulfide linkages. That is also correct. Is a non-fibrous structural protein. It is a fibrous structural protein. So option D is the incorrect one or the false one. So option D is the answer. The next one. Charcoal rot, also known as dry weather wilt, is caused by. So it is caused by Macrophomia fasciolina. Answer is option A. Then electroporation is an experimental technique adopted in. So it is used in genetic engineering studies for transformation of plant explants or tissue. We use electroporation technique. So answer is option B. 2,4-dichlorophenolic acid is generally used as. It is used as herbicide. 2,4-D. It is a herbicide. Then the C4 plants are different from C3 plant with reference to the type of end products of photosynthesis, number of ATP molecules that are consumed in preparing sugar, types of pigments involved in photosynthesis, substance that accepts carbon dioxide in Calvin cycle. So in C4 plants, every carbon dioxide molecule has to be fixed twice. So these plants need more energy for synthesis of hexose sugar than C3 plants, in which carbon dioxide has to be fixed only once. So 18 ATP molecules are required by C3 plants for synthesis of 1 molecule of hexose sugar, whereas 30 ATP molecules are required by C4 plants for the same. So the number of ATP molecules that are consumed in preparing sugar. So C4 plants differ from C3 plants with reference to number of ATP molecules that are consumed in preparing sugar. Then match the following. Rockweed ficus. Answer is option C will be correct because only C is having P as 4. Okay, we'll see the others also. Gulfweed, sargassum, reindeer moss, it is cladonia, and Irish moss is chondrus. So the option is P4, Q3, R2, S1. Okay, coming to the next question. Amphidiploid has played a major role in the production of new species of Brassica. The chromosome number of Brassica junctia, an amphidiploid that is produced by crossing Brassica nigra with Brassica campestris. So they have given the 2n, that is diploid chromosome number of nigra and campestris. It was used for producing the amphidiploid Brassica junctia. So the chromosome number of the amphidiploid is 16 plus 20, that is 36. Answer is option D. Then TikTok complex is associated with import of proteins into chloroplast. Answer is option B. Which of the following genera has 2 polar nuclei in its mature embryo sac? So Orchis is having 2 polar nuclei in its mature embryo sac. Then members of which of the following families lack endosperm? So Orchidaceae, they lack endosperm. Answer is option D. Which of the following class of genes does not act as transporters in plants? So it is AP2ERF that does not act as transporters in plants. Choose the right answer for the given statements on the location of photosystems in chloroplast. So statement 1. Pigment system 2 mostly occurs in the stacked region of thylakoids. That is correct. Pigment system 1 is predominantly present in unstacked regions of the thylakoids. That is also correct. So Option A is statement 1 is correct whereas statement 2 is false. Statement 2 is correct whereas statement 1 is false. Both the statements are true. Answer is option C. Apomixis is the type of reproduction which results in. So apomixis, it is defined as asexual formation of a seed from maternal tissues of the ovule, and they avoid the process of meiosis and fertilization, and then lead to embryo development. So embryo develops without meiosis and fertilization. So sorry. Development of plants without fusion of gametes. So apomixis, that is correct. A is correct, as well as development of embryo from tapetal cells. That is also correct. So answer is both A and C are correct. So coming to the next question, pectin, a component of cell wall, is stained by. So it is stained by ruthenium red. Answer is option D. Characteristic curling of root hairs in plants is associated with assimilation of nitrogen. Answer is option C. In a single, if a single somatic cell in a multicellular organism harbors a mutation, it is usually inherited in the next generation, has no phenotypic effect, causes cell death, creates hundreds of genetic alterations during DNA replication. So it is a single somatic cell in a multicellular organism that harbors a mutation. It will not have any phenotypic effect. Answer is option B. Then alpha D glucose and beta D plus glucose they are called anomers. So anomers are cyclic monosaccharides or glucosides that are epimers differing from each other in the configuration of carbon 1 at position 1. If they are aldoses, or in the configuration of carbon 2 if they are ketoses. So alpha D glucose and beta D glucose are anomers. Then next one. Match the acronym listed in the first column with the corresponding expanded form present in the second column. So CRISPR. CRISPR is clustered regularly interspaced short palindromic repeats. P3. So C and D we will not take into consideration. We will see A and B because they both have P3. Then RNAi, it is RNA interference, Q1. So the option naturally goes to B. Okay, then TALEN, it is transcription activator like effector liase, and miRNA is microRNA, S5. So B, option B is the correct answer, P3, Q1, R6, S5. So next question. So coenzyme is vitamin, P2. Then competitive inhibition. Competitive inhibition is succinic dehydrogenase by malonic acid. Trypsin is alkaline pH, Km and Bmax is Michaelis-Menten, and alpha helix is right-handed, T4. So the answer is option C, that is P2, Q3, R5, S1 and T4. Sorry, please excuse me. How many EDTA molecules are required to make an octahedral complex with Ca2 plus ion? So only one EDTA molecule is required to make octahedral complex with a Ca2 plus ion. Then which one of the following pairs is not matched? Magnesium and chlorophyll, copper and cytochrome, manganese and nitrogenase, molybdenum and nitrate reductase 1. So it is manganese and nitrogenase. It is not manganese, it is molybdenum and nitrogenase. Then next one. A student wanted to prepare 2500 ml of buffer containing 100 millimolar sodium acetate at pH 4 containing 0.01% NaCl. He has the stocks of 2 molar sodium acetate buffer pH 4 and 10% NaCl solution. What volumes of the given stock solution he has to mix in distilled water for preparing 2500 ml of the above reaction buffer? So you are having 2 molar sodium acetate buffer at pH 4 and you want 100 millimolar sodium acetate at pH 4 for 2500 ml. So M1V1 is equal to M2V2. So 2 molar is into X, that is V1 is equal to how much we are having, that is 100 into 10 raised to minus 3 molar into 2500 ml. So X will be around 125 ml. So 125 ml of sodium acetate at pH 4 is required. Then you are having a stock of 10% NaCl solution and how much, that is 0.01% NaCl do you require. So 10% from that how we have to take to prepare 0.01% of 2500 ml, that is 2.5 ml of NaCl. So you require 125 ml of sodium acetate and 2.5 ml of NaCl. So the answer is option A. Then next one, identify the monounsaturated fatty acid given below. So linoleic acid, it has got 2 double bonds, so it is not monounsaturated fatty acid. Then arachidonic acid, linolenic acid and oleic acid, you will find that oleic acid is the monounsaturated fatty acid. Okay, so the answer is option D, that is oleic acid. Match the cell organelles in group 1 with their functions listed in group 2. So peroxisome is for detoxification, P2. Mitochondria is for proton gradient formation, Q3. Ribosome is for protein synthesis, R4, and leucoplast are for storage of starch granules. So answer is option C, that is P2, Q3, R4, S1. Then the basis of precipitation of proteins by ammonium sulfate is best described by which of the following statement? So what does ammonium sulfate do to precipitate proteins? So ammonium sulfate, it binds to water molecules, making them less available for hydration of proteins. So that is how it precipitates the proteins out of the buffer solution. So answer is ammonium sulfate binds water molecules, making them less available for hydration of proteins. So answer is option D. Then next one, match the following diseases with pathogen causative factor. Creutzfeldt-Jakob, it is caused by prion, so P3. And pseudocytosis, it is caused by fungi. Legionnaires disease is caused by bacteria and rabies is caused by virus. So answer is option B. Then next one, one student from Kozhikode district of Kerala got his admission in Wuhan University, China for his PhD. He travelled to Wuhan University during first week of January 2020 to start his semester. Within 1 week of his arrival in Wuhan, he became sick and consulted University Health Center. Based upon his symptoms, the doctor immediately suggested him for Vidal test. His Vidal test report was positive. Which disease or pathogen he was suffering from? So Vidal test disease is for typhoid, and he was positive for Vidal test, that means he is suffering from typhoid. Answer is option C. Then next one, which of the chemical formula belong to valeric acid? So it is CH3, CH2, 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 COOH. That is the formula for valeric acid. Answer is option A. Then one scientist has given genomic DNA sample of a plant species to his research scholar and asked him to amplify a specific gene of interest XYZ using the available components in the lab. As suggested, he mixed all the components and performed the experiment, which was finally failed. Upon investigation, his supervisor realized that his students forgot to add one very important component to the tube. His student mixed the following in the tube: genomic DNA, pair of primers, 4 types of DNTPs, buffer and water. Which component his student forgot to add in the tube? So here what is happening is a scientist gave a genomic DNA sample and asked his student to amplify a specific gene of interest, that is XYZ, using the components available in his lab. The student mixed the components. What and all did he mix? He mixed genomic DNA, primer for pairs, 4 types of DNTPs, buffer and distilled water. So from these components itself we know that he is performing a PCR reaction, and moreover from the question it is again clear that the scientist asked the student to amplify a specific gene of interest. So naturally he will be performing PCR only. So upon investigation, his supervisor, but he didn't get the result. Okay, the student didn't get the result. So and the supervisor investigated what his student forgot to add, and he found that his student forgot to add one very important component into the tube. So what is that component? Out of all these components that is given here, Taq polymerase is missing in his PCR reaction. So naturally when Taq polymerase is missing, he will not get the amplified product. So answer is option B, that is Taq polymerase. Then what is the term amphipathic means? Amphipathic means a chemical compound or biomolecule which possesses both hydrophilic and hydrophobic properties. So answer is option C. Then when a protein containing tyrosine and tryptophan is warmed with concentrated nitric acid, it has turned to yellow color because of nitration of aromatic ring. This test is called as xanthoprotein test. So answer is option A. Okay, coming to the next question. Which of the following human disease is caused by helminth? So it is trichuriasis that is caused by helminth. When ethylene is treated with sulfur monochloride, a toxic product is obtained. This product is mustard gas. Answer is option D. In plant biology studies, some of the abbreviated terms are used which are listed in column A. Match its best combination present in column B and select the correct answer. XA21. So that is rice disease resistance gene, AP5. SMA1, that is a restriction endonuclease, Q4. DAPI, it is a type of fluorescent dye, and RFLP is restriction. Okay, it is restriction fragment length polymorphism. It is DNA based marker technique. So answer is option B, that is P5Q4R2S3. The next one. Which of the following is considered as a secondary metabolite of higher plants? So it is carotenoid that is considered as a secondary metabolite of higher plants. Then morphine was initially isolated from Papaver somniferum. Answer is option A. Ocimum tenuiflorum belongs to. So Ocimum belongs to Laminaceae family. Answer is option B. Plant tannins are polyphenolic compounds. Answer is option A. Then match the common names of the plant listed in the left panel with their scientific names from the right panel. Choose the correct answer. So finger millet. Finger millet is Eleusine coricana. That is option P3. Foxtail millet, it is Serratia italica, Q4. Pearl millet is Pennisetum glaucum, L. R2. And oat is Avena sativa, that is S1. So the answer here is option B, that is P3Q4R2S1. Then again match the following. So lysine is positive charge R group. Lysine has got a positively charged R group. So P2. Then aromatic amino acid, it is tyrosine, Q4. Cysteine is sulfur containing amino acid, R5. Arginine is guanidino group S1. Alpha helical structure is T3, alanine. So the answer is option A, that is P2, Q4, R5, S1, T3. Then next one. Match the following and choose the correct combination. Rho and RAC proteins. They are GTP binding proteins. So P3. RB is tumor suppressor gene. MAP kinases. That is serine threonine kinase. Answer is R1. And BAD-S4, apoptotic factor. So answer is option C, P3Q2 R1S4. Then next one. Match the following and choose the correct answer given below. Inbred. Inbred is cross-pollinated plant progeny. P4. Pure line. Pure line is self-pollinated plant progeny. Clone. Clone is vegetatively propagated plants. And hybrid. Hybrid is first filial progeny. So answer is option C, P4Q3 R1S2. Next one. In E. coli, 4 HFR strains donated the following genetic markers shown in the order donated. That is, strain 1 donated QWDMT, strain 2 AXPTM, and strain 3 BNCAX, and strain 4 BQWDM. All of these HFR strains are derived from the same F plus strain. What is the order of these markers on the circular chromosome of the original F plus? So I have given it in the next slide. So here there are 4 HFR strains that donate genetic markers. So all these HFR strains are derived from the same F plus strain, and they have asked for the order of these markers in the circular chromosome of the original F plus. So these are the strains, that is strain 1, strain 2, strain 3 and strain 4. You will see that they have been arranged, the markers have been arranged like this, okay, on the strains. So when you join all these things, see here QWDMT. Now where does T come? T comes here. Okay, that is AXPTM. So TM. So after T and M, it should be PXA. So the next is XA. XA is here also in strain 3. So after XA, you will get CNB. Okay. And in the fourth strain, CNB, there you have got the B portion along with QWD and M that belongs to strain 1. So the order of this particular gene is QWDMTPXACNB. Okay, so we'll go to the next question. Then miRNA has been shown to play a significant role in gene expression. Some miRNAs induce gene silencing by binding to mRNAs and inducing inhibition of translation. On the other hand, there are miRNAs that bind to mRNAs and activate their degradation. The following characteristics can be applicable to the miRNAs that inhibit mRNA translation. So, miRNA is partially complementary to a region of target mRNA in the 3' UTR. That is correct. Then miRNA always base pairs with full mRNA and an AU-rich sequence. That is not correct. miRNA base pairs with mRNA through 6 to 7 nucleotide at its 5' end, referred to as seed sequence, as well as a few additional bases elsewhere. That is correct. So 1 is correct, 3 is also correct. Then miRNA is always partially complementary to the conserved sequence of the target mRNA. That is also not correct. So 1 and 3 are correct. So answer is option B, 1 and 3. Coming to the next question. When the dihybrid ratio 9:3:3:1 gets modified to 9:3:4 due to gene interaction, then it is referred to as recessive epistasis. That is when recessive alleles at one locus mask the expression of both dominant and recessive alleles. At another locus, it is known as recessive epistasis, and you will get the formula, that is dihybrid ratio, that is 9:3:4 due to this particular gene interaction instead of the usual Mendelian dihybrid ratio, that is 9:3:3:1. The next one, promoter is upstream DNA sequence of a gene which is recognized by RNA polymerase in order to initiate transcription. Answer is option B. That's a direct question. Then, regarding the relationship between two organisms in an ecosystem, match the following. Commensalism. So in commensalism, one organism benefits but the other is unaffected. So P3. Then mutualism, both organisms are benefited. And parasitism, one is benefited, other is harmed. And amensalism, one impeding the success of the other. So answer is option C, that is P3Q1R4S2. Next one. Mutation in a gene X in Arabidopsis thaliana result in more number of lateral root formation. Which one of the following is the correct statement? The gene product act as positive regulator of lateral root formation. That can be a correct statement. Okay, we'll see. The gene product act as negative regulator. It doesn't act as negative regulator because you get more number of lateral roots. The gene product is most likely to be involved in lateral root formation. That is also not correct. It's not likely to be involved in the lateral root formation. That is also not correct. The gene product neither promotes or reduces the lateral root development. That is also not correct. So the gene product acts as a positive regulator of lateral root formation, and positive regulation, it is any process that increases the frequency or rate of that particular gene expression. So answer is option A. Identify the corresponding target sites for the following restriction endonucleases. So EcoR1 is having GAATTC, P3. BAMH1 is GGATCC. So Q1. Hind3 is AAGCTT. And NOT1 is GCGGCCGC. So answer is option D. Then next one, match the scientist names given in panel A with the corresponding contributions given in panel B and choose the correct answer. So H.J. Muller for mutagenic effect of X-rays in Drosophila. C. Bridges, he was a non-disjunction proof that chromosome contain genes. And Barbara McClintock or B. McClintock for discovery of mobile genetic elements or transposable elements. And T.H. Morgan for discovery of sex linkage. So answer is option A, P3Q1R4S5. Then, which of the following can form a part of biosensor? So biosensor is a device which uses a living organism or a biological molecule, usually enzymes or antibodies, to detect the presence of chemicals. So answer is which of the following can form part of a biosensor is enzyme, antibody as well as a lipid. So answer is option D, 1, 2, 3 are correct. The next one, match the aneuploid condition of a diploid organism given in panel A with corresponding description given in panel B and choose the correct answer. So nullisomy. Nullisomy is loss of both members of a homologous pair of chromosome. P4. Monosomy. Monosomy is loss of a single chromosome. Q2. Trisomy. Trisomy is gain of a single chromosome. R1. And tetrasomy is gain of two homologous chromosomes. S3. So the answer is option B, P4, Q2, R1, S3. So coming to the next question, which is the set of forward and backward primer pairs of below given sequence? So they have given a sequence and they have asked what is the forward and the backward primer. Okay, I will do it in the next. So this is the sequence that they have given. This written from 5' end to 3' end, and this is the complement of that sequence, this second sequence, that is 3' end to 5' end. I have written the complement also of that particular given sequence. Now your forward primer, it will bind here. This is the forward direction, that is 5' to 3' end. Okay, so this is the forward primer. So it will be complementary to this particular sequence in the 3', 5' direction. So it is ATC, GTG, CT, ATT, CGT, CGA. And that is same as the sequence of this particular first strand that is given in the 5', 3' direction. So it is same as that of the given strand. So this will be your forward primer. So this particular sequence is the forward primer, that is similar to the top strand. Okay, so that is the forward primer. Now for the reverse primer, that will bind over here, that will be complementary to this particular sequence but in the 5' to 3' direction. So this is the 3' end, so here we will write the 5' end. So complementary to this C, G, A, G, T, C, G, A, G, T, C, A, G, C, T, T, A. That is the reverse primer. So 5' to 3'. They have given in the options in the direction 5' to 3' direction. So it will be this. So this will be the reverse primer and this will be the forward primer. So the answer, I think it is option B. Then next one, homeobox sequence is. So it is a conserved sequence which regulates development of many animal species. Answer is option C. Then match the below research institutes with places. International Crop Research Institute for Semi-Arid Tropics. So it is situated at Hyderabad. So P3. Then National Institute of Virology is in Pune, Q4. Indian Institute of Horticulture Research is in Bangalore, R2. Institute of Forest Genetics and Tree Breeding, that is S1. So answer here is option A, P3, Q4, R2, S1. So coming to the next question. Ribulose 1,5-biphosphate carboxylase oxygenase is also called as carboxydimutase. Answer is option D. Then next one. Which of the following are involved in the derivation of induced pluripotent stem cells? Now induced pluripotent stem cells or iPS cells or iPSCs are a type of pluripotent stem cells that can be generated from adult somatic cells such as skin fibroblast or peripheral blood nuclear mononuclear cells by genetic reprogramming or by forced introduction of reprogramming genes. Now these reprogramming genes are OCT4, SOX2, KLF4, c-MYC genes. So the answer here is which of the following are involved in the derivation of induced pluripotent stem cells from somatic cells is option D. These are the reprogramming genes. Okay, nitrogen fixation is not carried out by higher plants. Answer is option B. Now next one. In Mendel experiment, the seed character SS is completely dominant over the dented seed character. smallest. If the characters for height were incompletely dominant such that the capital T are tall, capital T, small t are intermediate, and small t are short, what would be the phenotypes resulting from crossing of spherical seeded short plants to dented seeded tall plants? So I have given that answer in the next. So this is the spherical seeded they have given in the question. That is spherical seeded short plant crossed with dented seeded tall plant. So SS small t, smallest, capital T. You will get spherical seeded plant, but because this small t and capital T they show intermediate height, you will get spherical seeded and intermediate height plants. So all of them will be spherical seeded and intermediate height plants. So answer is option D. Then next one. Which of the following pairs is not correctly matched? So niacin is pellagra, vitamin B12 is pernicious anemia, vitamin C is scurvy, and vitamin B6 is beriberi. So vitamin B6 beriberi, it is caused by vitamin B1 deficiency, that is thiamine deficiency, not by B6. So the not correctly matched is option D, that is vitamin B6 beriberi. Then maize seeds become viviparous when they are deficient in abscisic acid. This is also a repeated question. Then, which of the following is not present in plant cells? So, centrioles are present in animal cells, they are not present in plant cells. Then, glycolysis is one of the important metabolic pathways to convert glucose to pyruvate. Identify the specific glycolysis products given below. So, glucose 6-phosphate, it is a glycolysis product. Acetyl-CoA is not a glycolysis product. Then glyceraldehyde 3-phosphate is again a product of glycolysis, and 2-phosphoglycerate is also a product of glycolysis. So the answer is option D, that is 1, 3 and 4. So coming to the next question. Bulbosum method has been used in barley to generate. It has been used in barley to generate double haploids. Answer is option A. Then which of the following photoreceptors has terene as chromophore? So it is cryptochrome. Answer is option B. Then next one. Match the plant growth regulator from left panel to their effects listed in the right panel. Mark the correct answer. So auxin. Auxin inhibits outgrowth of axillary buds. P3. Cytokinin, they prevent senescence. Gibberellic acid, they break seed dormancy, R1. And ethylene, it promotes senescence of flowers, S2. So answer is option A, that is P3Q4 R1 S2. Then which enzyme is involved in epigenetic inheritance? So So it is histone methyl transferase that is involved in epigenetic inheritance. Answer is option D. Then flowers represent a complex array of functionally specialized structures that differ substantially from the vegetative plant body in form and cell types. Following are statements made regarding floral meristems. Which combination of the above statement is true? So floral meristems can be distinguished from vegetative meristems by their larger size. That is correct. The increase in size of the meristem is largely a result of increased rate of cell division in central cells. That is also correct. The increase in size of the meristem is due to larger size of the cell, which in turn result from rapid cell expansion only. That is not correct. Here we have told that increased rate of cell division in central cells. Then a network of genes control floral morphogenesis in plants. That is also correct. So the combination that is correct is answer A, that is 1, 2 and 4. Then zDNA is one of the many possible double helical structures of DNA. However, it is left-handed DNA helix. Answer is option C. Match the terms listed in panel A with the description indicated in panel B. Choose the correct answer. So xerophyte. Xerophyte is plants that grow in places with scanty water. P2. Hydrophyte. Hydrophyte is plants grow in water may rooted in the mud. Q4. Mesophyte is plants that grow under average conditions of temperature and moisture. R5. And halophyte is plants adapted to saline soil. So answer is option B, that is P2, Q4, R5, S3. Then next one. The diploid chromosome number of Siam maize is 2n is equal to 20. The number of chromosomes and DNA molecules that are found per cell when this original cell progresses through G2 phase of mitosis is. Now the 2n number is 20. So G1 phase will have 20, S phase it will be doubled. So 20 will become 40. So it will be carried over to the G2 phase. So G2 will have 40. So the number of chromosomes and DNA molecules that are found per cell. So the number of chromosomes will be 20, but the number of DNA molecules will be 40 in the G2 phase of mitosis, because G2 comes after the S phase, and S phase there is replication of DNA. Then the cluster of oxygen evolving complex in photosystem 2 contains Mn4O5Ca. So answer is option D. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.